Hello dear students of class 8, this is Mamta Verma once again brings a video for you. In my previous video I have taught you the chapter microorganism. That was its first part. This is the second part of this chapter. So let's explain. I will explain from page number 32. Before this, I was explaining about the beneficial microorganism. So we'll be continue with that beneficial microorganism. Some microorganisms also help in agriculture. Plants need a large amount of nitrogen to prepare protein in their bodies. Although 78% nitrogen is present in the atmospheric air, plants are unable to use it directly. They can take up nitrogen only in dissolved form as nitrates. Some blue-green algae and rhizobium bacteria that live in root nodules of leguminous plants help in the conversion of atmospheric nitrogen to nitrates. This is called nitrogen fixation. Thus, these microbes help plants get their nitrogen content from soil and are known as biological nitrogen fixer. So, though there are maximum amount of nitrogen in our atmosphere, but plants are unable to fix it directly from air. So, these microorganisms first convert these nitrogen into nitrates and nitrites and make them able to absorb by the plants. Next, in the cleansing of the environment, as you have studied from your childhood that most of the saprophytic microbes such as bacteria and fungi feed upon the dead organic matters like dead plants or animals. They break down or decompose complex organic substances to simpler one and are therefore also known as decomposer. Through the process of decomposition, organic nutrients from the dead organisms are released into the soil and are taken up by the plants again. Thus, decomposers serve two important functions. First, they prevent our earth from being converted into a heap of dead organism. Second, they maintain the flow of energy in the environment by releasing nutrients into the soil and allowing them to enter the food chain. They also add humus to the soil, which makes it fertile. Now, some harmful microorganisms. Harmful microorganisms means the microorganisms that harm to us. So you can understand that disease producing organism or microorganism can be the harmful microorganism. So diseases that are caused by microbes could be transformed from an infected person to a healthy person. Such diseases are known as communicable diseases, that is caused by microbes. So diseases are of two types, that is communicable diseases and deficiency diseases, but only communicable diseases can be caused by these microorganisms. They may be spread by direct contact with the infected person. Some pathogens do not enter the body at person directly. They are carried by other animals or insect known as carriers, or this is also called vector. For example, malaria is caused by the protozoan called Plasmodium vivax. This is carried by the female Anopheles mosquito. When a female Anopheles bites a healthy person, the Plasmodium is transferred to the body of that person. So here we can say that Anopheles mosquito is the vector of that disease, the disease called malaria, and the main pathogen, that is disease causing organism or microorganism, is Plasmodium, which is a protozoan, that is protozoa. Protozoan is the singular form, protozoa is the plural form. So here is some name of some diseases, its pathogen and mode of transmission in a tabular form. So you have to learn all these diseases. Next, prevention of diseases caused by pathogens. So, you have mostly heard that prevention is better than cure. That means we have to prevent some diseases. It is mostly better than cure. That means if some disease is caused to your body, that it will most harm to your body, but if we prevent them to enter our body, then it is much better. So the some following way help prevent diseases caused by pathogens. Proper vaccination of children should be carried out. Personal hygiene should be practiced properly. Cleanliness, that is called personal hygiene. One should wash fruit and vegetable before eating. Food should be kept covered. One should avoid direct contact with an infected person. Mosquito and insect repellent should be used. Mosquitoes and insect breed in stagnant water. Necessary measures should be taken to ensure water does not collect in a place. Most places has the waterborne diseases, malarial parasite born in the stagnant water, that is fixed waterlogged places. So we need to remove this type of places nearby our houses. Plants can be prevented from acquiring various diseases by spraying them with pesticides and fungicides from time to time. Proper vaccination of pet animals should be ensured. Now, spoilage of food. If food is not stored properly, it may get infected by the pathogens. These pathogens may not be harmful themselves, but they produce toxic substances in food by oxidation. Such food can be identified by a foul smell, a change in color, and unpleasant taste, and can no longer be consumed. If this food is eaten, it could cause headache, dysentery, vomiting, fever. Some infections may also prove to be fatal. That we call it food poisoning. So, next, spoilage means the stealing of food. You mostly seen during the summer time, if some dal, that is pulses, prepared dal already, all we eat every day in our diet. So if we keep it till evening, then it has a foul smell. So it is stale or spoiled. It becomes spoiled. So this is caused due to the growth of microorganism in this pulse. And the microbes that grow in that food products are Salmonella, Staphylococcus, and Aspergillus that develop in the food and spoil it. Then we need to preserve these foods by certain methods. So the use of physical or chemical methods to stop or slow down the growth of microorganism to prevent food spoilage is called food preservation. So there are certain methods. I will explain one by one. So first one is controlling temperature. Pasteurization. It is a method involves heating of milk to about 70 degrees Celsius for around 15 to 30 seconds and then cooling it rapidly before it is stored. So this is developed by, this pasteurization was developed by Louis Pasteur, and so in this process milk is preserved. Second one is sterilization. It is a technique of killing germs by heating the food items to a temperature of 100 degrees Celsius or above. 100 degrees Celsius or above temperature is sufficient to kill microbes. So we often suggested by doctors to drink boiled water. So first we should boil water till 100 degrees Celsius and then after cooling it we should drink it. Next one is by using preservatives. Some preservatives check the growth of microbes when added to food. These are called preservatives. Such as salt, sugar, oil, vinegar are common substances in pickles. Generally in pickles we made at home, mostly salts are added. In jam, sugars are added to prevent the growth of microbes. One more thing is that in addition to these methods, precautions to prevent the growth of microbes are also taken while packing food. Since the presence of oxygen promotes the development of pathogens, food items are packed in an atmosphere that is rich in nitrogen content rather than oxygen. Foodstuffs are also vacuum packed for similar reason, such as you eat the chips packet which is full of nitrogen to prevent the stealing of chips. You have taught right now the fixation of nitrogen. So how nitrogen is fixed, and this fixation of nitrogen is called nitrogen cycle. So nitrogen fixation may takes place by three processes: biological fixation, atmospheric fixation, and industrial fixation by various chemical process in the industry. First one is biological fixation. It is carried out by the symbiotic bacteria Rhizobium residing in the root nodules of the leguminous plants. They convert the free nitrogen into nitrates, which get mixed with the soil after decay of these plants and are subsequently taken up by other plants. Blue-green algae are also capable of performing the same function. Besides, some free-living soil bacteria are also involved in conversion of other forms of nitrogen like ammonia to nitrites and then nitrites to nitrate. So it is a multi-step process for the conversion of nitrogen to nitrates, and also this. So here we can say one more example of symbiosis, that is the living of bacteria with root nodules of the leguminous plants. So this is also another example. We already almost even often give the example as only lichen, but here is another example of symbiotic association. The process of conversion of ammonia to nitrites and subsequently nitrites to nitrate is called nitrification. The bacteria involved in nitrification are Nitrosomonas and Nitrobacter are called nitrifying bacteria. Atmospheric fixation takes place because of atmospheric lighting. Lightning strikes cause atmospheric nitrogen and oxygen to react with each other to form nitric acid. This nitric acid comes down along with rainwater and forms nitrates on reacting with limestone. Industrial fixation is carried out in various industrial processes. Compounds like nitric acid and ammonia are produced in industries. These are converted into different fertilizers like urea. These fertilizers enrich the soil with the soluble form of nitrogen. We often see that during the agricultural practices, the farmers give a white round type of substances to the crop field, and again spraying some DDT. So this white colored substance is urea. And why this nitrogen is so important for the plants? Because nitrogen is the main substance for growth of the plants. So nitrogen is necessary to take by the plants. And what is nitrogen cycle now? Nitrogen cycle is a multi-step processes, and different steps are involved within this nitrogen cycle. First one, you have right now studied about the nitrogen fixation, that is atmospheric nitrogen is converted into nitrates by nitrifying bacteria in the soil through the process of nitrogen fixation. Second one is assimilation. Nitrates are taken up by the plants through their roots to prepare amino acids, protein and nucleic acid. When plants are eaten by animals or humans, the nitrogen passes into their bodies through the food. So assimilation is the process when nitrates are taken up by the plants. It's a breakdown into many substances like amino acids, proteins and nucleic acids. When these plants are eaten up by the animals or humans, the nitrogen passes into their bodies through the food. This process is called assimilation to convert it into the energy. Next one is ammonification. This nitrogen is added back by the animals and plant to the environment in combined form as organic compounds. When plants or animal die, these organic form of nitrogen are released into the soil, and certain bacteria present in the soil convert the organic nitrogen to the ammonia. This process is called ammonification. Again, this ammonia is going back to the atmosphere. How? So this process through this nitrogen cycle. And last one is denitrification. Some special bacteria present in the soil help to convert ammonia into the inert nitrogen. This process is called denitrification. So these are the important steps, you have to learn it. These bacteria are called denitrifying bacteria. Pseudomonas and Clostridium are the example. Bacteria are mainly involved in denitrification, thus the cycle is complete and free nitrogen again enters the nitrogen cycle. So what is nitrogen cycle? That is the circulation of nitrogen from atmosphere to the plant soil, and from soil to the plants and then animals, and again going back to the atmosphere. This is called nitrogen cycle. Okay, now this chapter is completed in 2 parts. So thank you.